I see a pretty good turnout today, so I'm really happy to see. So it's our great pleasure to have Professor Mahadevan from Harvard come here, give a distinguished lecture. Don't ask me to pronounce his first name. In fact, I don't even know he himself knows how to pronounce it. Anyway, so I actually, of course, I heard quite a bit about Professor Mahadevan, especially I have a little bit of passion of origami, and my kids is now really into it, and so they were actually really looking forward to get a chance to see his origami. And of course, he told me because they're too big, and so they were a little bit disappointed yesterday, okay? So anyway, many of you obviously know the MacArthur Fellowship, which is also called the Genius Prize. And Maha is actually, without any further sort of mentioning of the other awards like Royal Society Fellow and this sort of thing, let me just say he is actually one of the MacArthur Genius Award recipient. Okay. And his research interest is extremely broad. I think you graduated from Stanford in '95 as applied mathematics. But his position at Harvard spanned 3 departments. Okay. Evolutionary biology, is that physics, and applied mathematics. Okay. And to give you a glimpse of the depth of his work, he gave me 3 titles. One of which is the one you're going to hear. I actually prefer his title with origami, but because I might be the only one who is interested, so we choose a title which spans life science, mathematics, and physics. But I'm sure he has so many other good ideas. I think it would be a shame if you don't have a chance to interact with him a little bit more in depth, because he is a man with so many good ideas and very broad interests. It's a Renaissance man. I don't think I need to say too much, okay? And so without further ado, let's just welcome Professor Mahadevan.
Well, thank you very much, Dean Yang, for this very generous introduction. From now, it's going to be downhill, I'm afraid. But it is a Friday afternoon. I think I can't quite remember. Say I'm 12 hours out of sync, and so 12 hours out of sync is the worst possible situation. Doesn't matter which way it is. So I hope I'll try and amuse you for the next hour or so. And I want to tell you about some problems that I've been thinking about for almost a decade now, and it was all inspired by this book, which if you have not seen, I encourage you, especially amongst younger people. On growth and form, whose centenary was celebrated by a number of events last year. And this book was written by this remarkable man, Scottish scientist, amateur mathematician by his own account, perhaps much more than that. He was trained as a biologist. He loved words. He loved languages. He was an expert in Greek and Latin. And Peter Medawer, very famous British academic, said that in fact this book is one by any stretch of the imagination probably the most literary masterpiece ever written in science. And this is Darcy Thompson. He had eccentric habits, including essentially walking around Dundee with a parrot in his arm. And in this book, all of which is essentially an exhortation asking the scientists of his time to essentially think about biology from a more physical, mathematical point of view. And he says the whole book is an introduction. It's 1,000 pages. It's Dover, so it's available even now. He says right at the beginning of the book, the problems of form are in the first instance mathematical problems. The problems of growth are essentially physical problems. And the morphologist is ipso facto a student of physical science. And so that's the mandate from which I'd like to try and approach the problem of growth and form inspired by this title of the book itself. And as I hope it'll become clear over the next hour, I want to somehow use the idea that when you think about growth and form, you're fundamentally thinking about geometry, you are thinking about geometry inspired by biology, and the ideas that we need to try and understand them come from physics and from mathematics. Okay. Why do we care about form? We care about form because in architecture, for example, the famous architect Louis Sullivan said that form ever follows function. But in fact, in biology, perhaps function also ever follows form. And I'm going to give you some examples of this, things which are very immediately obvious to you, something that I'll come back to. Your brain is very folded cortex. And that folded cortex allows you to have a very large number of connections in a relatively small volume, because if I fold it up, then I can essentially have substantial amounts of area. So that's for information processing at high speeds. Your lungs are folded, and if you open up your lungs, you can essentially cover a substantial fraction of a tennis court. And again, it's the same thing. You have a folded lung so that you essentially maximize the amount of area now for transfer of gases. And your intestine is similarly folded, and I'll talk about the intestine as well. So very, very often in biology, we have function and form ever so tightly linked to each other. And it's not at all clear, because everything happened through evolution, whether it's one way. It's not just that form ever follows function, but function follows form. And over time, these iterations gradually lead to the complex biological forms that we see around us. And we want to think about these problems, and we want to understand them. As in every field, we want to essentially do so using comparisons, because we don't want to just look at any one example, since it may not be representative of the whole class. If you were a biologist, you approach these problems in an organism by studying development, how the organism over its lifetime changed from a single cell to a complex multicellular organism like you and I are. If you're an evolutionary biologist, you want to essentially also now account for how this happened, not in one organism, but across organisms and over much longer time periods than that associated with the lifespan of an organism. And in particular, you want to understand the class of shapes and the class of forms. If, on the other hand, you think about this from perhaps the perspective of many people over here, and certainly the perspective that I we have tried to look at, then you want a theoretical description, and the theoretical description fundamentally must rely on laws of physics. And by models, I don't necessarily mean mathematical models, I also mean physical models, actual real physical models, which try to mimic some of what you see in biology, because biology is associated with the transmutation and transmogrification of matter in all its forms, and those forms of matter, of course, follow the laws of physics. And then if you can understand it in one or more examples, we want to use mathematics as a metaphor which allows you to translate from one to the other. So this is a tall, very tall mandate, and you'll see where we are in this process. Okay, so if you wanted to try and describe shape and describe form, of course, you have to describe it using the only language we know, which is mathematics. You can ask questions about this beautiful greenery that you see around over here, which has been just absolutely fascinating every morning when I get up over here, and when I'm awake at night as well, as it happened yesterday. So I want to describe, for example, the shape of objects, leaves, trees, shoots, and so on and so forth. And Euclidean geometry is not sufficient. I have to think about non-Euclidean geometry, because these objects are not flat. But that might not be enough. I might think about a different perspective, and I'm not going to be able to talk about all these things, but I want to give you a way. Every aspect of mathematics that you can imagine, you can effectively use to understand and describe shape. So you may be able to use different kinds of geometries. Non-Euclidean geometry is one example. Recently, we had the opportunity to use ideas from projective geometry to try and understand the shape of eggs, bird eggs. And we analyzed more than 50,000 eggs from a few thousand different species. And we used very simple projective geometry ideas, which in a lay setting boil down to understanding the asymmetry of the egg shape. So as you move further and further towards the right side, you become more and more strongly asymmetric. And as you go further and further up, you essentially become more and more elliptic. And those correspond precisely to the two projective geometry coordinates that I need to describe curves in the plane, it turns out. And you can ask me perhaps later or over the break at the end why this part is not occupied, and I can actually tell you something about why it's not occupied. Okay? So you can use different, and we do see different views of geometry all in biology. You could use conformal geometries to understand, for example, the shape of the brain. And I will tell you a little bit about that again towards the end of the talk. And here, I'm just basically showing you images where you take the surface of a brain, you essentially then deform it onto the surface of a sphere, and then you use the first few eigenfunctions of a Laplacian on the sphere to try and characterize where the folds are. And you can essentially do this in a numerical setting using conformal maps. Or again, something else that we've been playing around with is you don't have to think about conformal geometries, you can think about quasi-conformal geometries. And there's a reason that we want to think about quasi-conformal geometries in these systems, because conformal geometries, of course, map circles to circles, and that's problematic because in biology, and in fact in physics, we don't necessarily map circles to circles, we may map circles to ellipses. And if we map circles to ellipses, then I have to ask not only how much the area changed, I have to tell you something about the orientation, and finally I have to tell you something about the eccentricity. So I have 3 degrees of freedom at every point in the plane, which is perfectly amenable to thinking about quasi-conformal geometries. And so we've started to ask questions about how we may describe objects using these different geometries. We are hardly alone in this. There are lots of people involved in this. But there is a catch. There is an advantage and there is a disadvantage. The advantage is every one of these, again, very familiar to people over here, is capable of giving you extraordinarily beautiful and elegant mathematics. That's the plus. The negative is it might not be physical because we're just constructing these as clever mathematical ways of representing shape without necessarily thinking about how the leaf became non-Euclidean or how the egg is somehow amenable to projective geometry or how a wing of an insect became quasi-conformal or the brain became conformal. So the big problem is that it's unphysical. The big advantage is it's mathematically very beautiful. And so there has been, and in fact, Darcy Thompson's last chapter, his book's last chapter on growth and form focused on transformations of shapes from one to the other, in which he described some of this. And this is a very, very active area of some parts of applied mathematics. I won't tell you a lot more about this. Instead, I want to take a different perspective. And the different perspective is I don't want to only describe it mathematically. We'd like to try and make some predictions which essentially account for the physical laws underlying shape. And Darcy Thompson himself said a long time ago, 100 years ago, that cell and tissue are so many portions of matter, and it is in obedience to the laws of physics that the particles have been moved, molded, and conformed. So we want to basically see how physical law applies. And over the last 30 or 40 years in developmental biology and in molecular cell biology in general, there has been a revolution. And the revolution has been associated with our ability to essentially take apart all the molecular players and do some spectacular imaging and genetic manipulation at every level that you can care to essentially look at. So we now know in many model organisms, but increasingly in non-model systems as well, we know which genes are acting, what the gene regulatory networks are, what the different signaling molecules are, which signaling molecules are diffusible, which signaling molecules are on the surface of the cell. We know how cells move around, how they essentially create extracellular matrix, how they divide. We know that they apply forces on each other. We know that the extracellular matrix essentially plays an important role in this. And as we move up in scale, we eventually come to things which we have known for half a millennium. We know that if we think about these on the large scale or the meso scale, we have to think about systems which, of course, necessarily must follow the laws of Newton, force balance, and mass conservation. And as we go up in scale, the geometry becomes more and more apparent, and eventually we come to morphology. And so there's a problem over here, which is, where do we look at the system? Do we choose to look at the system at the molecular scale? Do we choose to look at the system at the cellular scale? Do we choose to look at the system at a multicellular scale? And of course, all of these are relevant, and I have to make a choice today to tell you about some aspects of these. And so I'm going to particularly choose to look at a multicellular scale associated with mesoscopic systems, precisely because we are interested in thinking about these large-scale flows and large-scale shape changes that you see in biology. Also because it turns out that as you go up in scale, there is a coarse graining which allows the models to become simpler and simpler, contrary to what you might assume. So we're not going to be interested in creating models where you're trying to solve a problem by essentially replicating a situation where 1 mile is equal to 1 mile. We don't want a scale model. We want a coarse grain, and we want to have a model where you have a small number of parameters and yet one which is amenable to experiment. Okay? So we're going to look at the mesoscale, the millimeter to the centimeter scale, and I've chosen to essentially tell you about 4 examples which are arranged in order of increasing dimension, if you will. So I want to tell you a little bit about one-dimensional growth problems. Then I want to tell you about two-dimensional growth problems. One-dimensional growth problem associated with the growing of a shoot or a root. Two-dimensional growth problems associated with the formation of leaves, and both these are associated with plants. And then I thought from a physical and a biological point of view, I'll also tell you a little bit about 2 plus epsilon dimensions, it turns out, and I'll explain what that means in a little later. This is essentially how a chick gut looks like early in embryogenesis. Your gut also looked something similar to this when you were in your mother's womb, about week 10 or 15 in gestation. And then finally, I'll end up telling you a little bit about a 3-dimensional problem, which is essentially the shape of the surface of your brain. All these systems are multicellular, and in all these systems, the cells can change in their number, in their size, in their shape, and in animals, also in position. So you have 4 fields which can essentially change in space and time. Okay, the number of cells in space and time, the size of their cells, the shape of the cells, and the position. In animal cells, animal multicellular tissues, position changes, but in plant multicellular tissues, cells don't move relative to each other. So you have only 3 fields available. And from these 4 fields, or 3 fields, depending on whether you're thinking about plants or animals, coupled with what's happening in the molecular scale, you see this tremendous range of shape. And the question is, how do we do this? 2 caveats. One, which is a favorite quote of mine, and perhaps a lot of people over here know, George Box famously said, all models are wrong, some are useful. I like one which is even better, perhaps, which Paul Ginsberg told me. I don't know who it's due to. He said, all models are right, most are useless. And so we have to be somewhere in the right part, probably in the left part of this figure, so that we actually are making sense. Okay, so compared to the descriptive mathematical perspective, the difficulty with physical models is potentially that there may be too many parameters in them, and therefore we may end up in this terrible mess where we don't really know whether we are right or wrong. On the other hand, if we are close to experiments, and I'll try and indicate how close we are, and you be the judge, there is a big advantage, because I can be essentially in a situation when I can be wrong in an interesting way, exactly like what George Box meant. Okay, so I want to start with the one-dimensional model associated with the growth and form of shoots. And if you have plants in your house, or if you've essentially observed all the wonderful greenery around over here, you've seen variants of what I'm showing you over here. You've seen that some shoots grow up very beautifully. This is Arabidopsis, a classical system, model system in plant biology. The shoot directly grows up, and it grows up for 2 reasons. It's growing up because it's negatively gravitropic. It wants to go against gravity, and it's also positively phototropic. It's essentially moving towards the sun. And it turns out that the sun is not important. How do you know? You can turn off the lights, and you can essentially do the experiment, and it will still go up. Okay? But this doesn't always happen. Even in the absence of light, sometimes the shoot grows up, sometimes the shoots grow down, sometimes they grow down and seems like they change their mind and start growing up. Okay? And so if you watch this, as I said, if you have house plants, I encourage you from now on to use a GoPro camera or something of that sort and do time lapse, and you'll see this whole thing happening in front of you. Okay? So the shoots sometimes grow down, sometimes they grow up, and you can ask, can you essentially make sense of this? And this is hardly a new problem. How can we make sense of this? I wanted to give you a little story that we've been involved in. It continues. I'll tell you a little bit about where we are. So this is a approximately one-dimensional object, and so mathematically I can completely characterize it if I tell you what the curvature of this little curve is in the plane. It can be in 3 dimensions. I'll focus on the planar problem. I'll tell you a little bit about the biology as well. So the curvature we denote by the rate of change of the angle relative to arc length, for example. And I will tell you about the biology. The shoot doesn't grow everywhere. Most of the growth occurs near the tip. And so there is a zone called the growth zone. It's a diffuse region. And these shoots grow only in that region. If you wait longer and longer, the paths away from the growth zone become more and more brown, more and more woody. All of you, I'm sure, noticed that. So gravity is sensed over the entire growth zone. It's not sensed at a point. It's sensed over a characteristic zone. And so now I want to raise a puzzle. So a very famous botanist more than 100 years ago suggested that a very natural way to understand how the shoot can essentially orient itself against gravity, he proposed effectively in words this mathematical equation. He said the rate of change of curvature is proportional locally to the deviation from verticality. Okay? It's a periodic function. Theta is a periodic function, so I want it to be proportional to sine theta. So what does it do? It says that as soon as the right-hand side is non-zero, then the shoot essentially turns, and it turns so that essentially it becomes zero, and then the curvature becomes zero, and it's growing negatively. But there's a problem with this, and the problem is indicated in this picture, but I'll essentially, either as you look at this picture, I'll wave my hands and show you what's going to happen. So I told you that sensing and growth doesn't just occur at a point, it occurs over an entire zone. So suppose this is my shoot, and the shoot is starting to become vertical. The first part to become vertical very likely is going to be the tip. But if the tip is vertical, the rest is not vertical, so the rest is going to start becoming vertical. But if the rest becomes vertical, then the tip is essentially gone past. And so now you're going to have a problem. Because if the tip is now gone past, then the tip is going to correct. But if the tip is going to correct, then the thing behind is going to get left behind. And so because I have a diffuse growth zone, this is not going to work for you. Okay, if it was a point sensing, point actuation, for those of you who know control theory, you have no problems. But as soon as I have a diffuse zone, this is not going to work. It's going to be very susceptible to noise, and you're going to get massive oscillations because you're never going to be able to essentially grow vertically. Surprisingly, that law stood until about 5 years ago, all right? And a couple of French physicists essentially proposed a different law, slightly modified. So René Bastien and his colleagues proposed a different law. And they basically said, you know what? I can add a term to this growth law. And I'm going to add this term. When I add this term, then it essentially takes care of everything. And the term in neurobiological language is proprioception. Okay? I know where my body ends and where the world starts. And hopefully we continue to know that so that I don't jam my fingers in the door and my toes in the door. Okay? So what they proposed was to say that the rate of change of the curvature is not only got a contribution which is associated with negative gravitropism, but it has another contribution associated with its ability to sense itself. Okay? And in biology, in botany, we know that's actually a fact. We know that there are cells in which there are amyloplasts, grains of starch, and these grains of starch settle, and the rate at which they settle and how much they settle is a function of the orientation of gravity. So in fact, we know at the cellular level it is possible for plants to sense gravity, just like it's possible for you and me to sense gravity using otoliths in my ears. And as they settle, I essentially can tell. And if you have a problem with your balance, that's associated fundamentally with your otoliths not necessarily working. So you add this term, and if you're a control theorist, you recognize that this kind of starts to look like integral feedback, okay? Because theta is the integral of the curvature. So this is a nonlinear version of integral feedback, except it's for a partial differential equation. It's not for an ordinary differential equation, okay? First thing. Second thing is, as soon as I write an equation like this, the left-hand side has dimensions of curvature per unit time. This is curvature, therefore gamma must have dimensions of 1 over time, and beta must have dimensions of 1 over length over time. And so the ratio of beta to gamma is a length scale. And that length scale is essentially telling you how strong gravity is compared to proprioception. If gravity dominates, it's just going to go straight up. It doesn't care. If, on the other hand, proprioception dominates, it wants to remain as straight as possible. So if you start out horizontal, it will try and remain that way. And so how do I know which is going to dominate? I have to worry about what is this length scale, which comes from the ratio of these two, compared to the length scale that's already present, which is the size of the diffuse growth zone. Okay? But now I want to change things and make them even more complicated. And here's the problem. Curvature comes in this equation on two sides, but it actually means something different. The curvature on the right side is the curvature that the plant senses. The curvature on the left side is the curvature that would be there if I removed all sensing. What is the natural curvature? Okay, so curvature essentially has two roles over here, a sensed curvature and an actuated curvature, because you should think of this, again, I'm going to use neurobiological language because it's very, very convenient to do so. This is the stimulus, it has two contributions, gravity and sense of self, and this is the response. And the response and the stimulus look like they should be the same, but they don't have to be the same, because the response is the intrinsic curvature, the stimulus is the measured curvature. And so there's a problem. And the problem is the response curvature here and the stimulus curvature are not going to be the same. And why are they not the same? They're not the same because the plant tries to grow, but as it tries to grow, it's subject to the physical world. And so in the physical world, it starts to sag. And if it starts to sag, then the sensed curvature and the actuated curvature are not the same. Okay? So I'm going to use an analogy to something that you're familiar with, which is your own posture. Okay? So I'm going to say that the actuated curvature, so your back, for example, or if you're a dancer or a tennis player or whatever it is, and you want to change your posture, you change your posture in response to some stimulus. In this case, the stimulus is associated with gravity and sensed curvature. And this curvature on the left side, the actuated curvature, is not the same as the curvature on the right side, because one is measured in the presence of the external world, in the presence of gravity, and what is measured over here, what is actuated over here, is what will be there in the absence of gravity. Now there's a problem, because I have one equation for two unknowns. I have to close the system. So how do I close the system? I have to bring back the physical world, I have to bring back gravity. And so not only do I have to control the posture of the plant, but I have to account for the postural mechanics of the plant. How do I do that? I do that by basically minimizing the total energy, and the total energy for an elastic system is the sum of the elastic energy, which tells you the deviation in the curvature from the rest curvature. The deviation is because of the presence of gravity, and then there is a constraint, and the constraint is equivalent in fluid mechanics to incompressibility. In elasticity, it's simply saying that the length is not changing. It's inextensible, up to a factor of growth. How do I know the relative ratio of gravity to elasticity? Well, I know that because there is another length scale in the problem. So if I'm very, very heavy, I'm going to stoop just because of gravity. If I'm extremely stiff, I can stay straight. So this parameter, G, characterizes the ratio of gravity to elasticity. And so again, in a neurobiology context, if you will, this parameter characterizes the interaction of the body with the environment, and this parameter B characterizes the interaction of the brain with the body. Okay? The brain is your ability to sense curvature. The body is how do you actuate curvature. Over here, gravity is the external world, so that's the environment, and the body is its internal elasticity. So with this system now, a PDE over here and a coupled to an ODE, which I can get by basically writing down the Euler-Lagrange equations, I have to solve them to understand how the system evolves. And there are two dimensionless parameters, one which characterizes the interaction of the brain and the body, and the other which characterizes the interaction of the body with the environment. And I'll just now show you some results. Okay? So you solve this problem numerically, as you have to, because it's nonlinear. And so you see that if the system is very, very light, so gravity is not playing a role, you recover the old result. So C and C star are the same. The brown simply corresponds to it becoming stiffer. If, on the other hand, it's very heavy, and it still wants to essentially be gravitropic, so it sags under its own weight, and it really can't basically do a whole lot. It's trying very hard, and eventually it's able to turn around and start to grow. So this is heavy and relatively sensitive. And this is yet another simulation for a different set of parameters where it basically sags and is not able to turn around because gravitropism is not strong compared to the weight. So proprioception dominates gravitropism. If proprioception dominates, it wants to be as straight as possible, and if it wants to be as straight as possible, it just sags down. So can we make sense of all this in the context of a phase diagram? So the Darcy-Thomsonian view is it's not enough, or the physics view or the mathematics view, it's not enough to know one example and see exactly how it works there. We want to ask, is the entire space of shapes available to us? And the answer is qualitatively yes. So here is the space of shapes in this two-dimensional phase diagram where one axis corresponds to how strongly you want to preserve your shape or how strongly you want to essentially respond to the environment. gravity, and the other axis corresponds to how strong the environment is. Is gravity very light, very weak, or you're very stiff, that's in this limit, or is gravity extremely super dominant, which means you're extremely floppy and very heavy. So as we move through this, we now start to make sense of what you see in your house plants. So plants which have the ability to go straight up are likely to be here, very strongly proprioceptive and likely very small. Large weeping willows, for example, and things of that sort, very strongly, very weakly proprioceptive, and very heavy, so that's over here. And then situations where it looks like the plant can't make up its mind, it goes down and then turns around and goes up. It's because there's a competition between gravitropism, proprioception, and gravity coming in through the body. So what's the message? So the message is perhaps, if nothing else, the fact that I can compress a lot of biological observations in terms of just a couple of parameters, one characterizing the sensitivity of the plant to different types of stimuli associated with its own shape, and stimuli associated with gravity essentially causing it to move negatively, away from gravity, and the other axis telling you about how the environment plays a role. And I think the message, at least for me, has been that you can't ignore the environment, you can't ignore the body, you can't ignore the brain. You have to worry about all three together. Okay, so that's my first example. I went through it in some detail. I'll now move to the others somewhat more fast. Second example, moving up in dimension, but limiting myself still to thinking about plants. So here is a set of images which inspired this work. And the question was, how do leaves ripple? So most leaves are not flat. Most leaves are rippled. In fact, if you ask yourself how easy it is to grow a flat leaf, you immediately realize it's extremely difficult. Without feedback, you can't do that. Because as soon as I grow a leaf, if it becomes large enough, the smallest wavelengths can grow, and if I have large leaves, very easily it can start to deform out of the plane. So without feedback, you can't essentially grow a flat leaf at all. Okay? We don't understand as a community how well and how one can grow a flat leaf. However, we do understand how easy it is to essentially grow non-flat leaves at different levels. So this paper, published about 15 years ago, has a wonderful title called The Genetics of Geometry. And in it, these authors, Nath and Enrico Cohen and colleagues, basically showed that they were able to manipulate the shape of a flat leaf of the snapdragon, a beautiful flowering plant, which is normally flat, to form this potato chip-like shape. And the way they were able to do this was by using a mutant, and this mutant essentially caused cells near the boundary to divide much more than the cells near the midrib. Okay, so if I have a flat sheet, and I have cells near the boundary which are dividing and have the same size, I increase the perimeter. And if I increase the perimeter, I can't remain flat. And so generically, I'll form something which is an approximation to the hyperbolic plane. So I'll form surfaces of negative Gauss curvature. And the shape of this, like a potato chip, is in fact not just an analogy, it's actually capable of being made mathematical. So if I take, how do you make potato chips? You take a potato, you slice it, you then take those slices which are flat and put them in boiling oil. Which parts dry out first? The boundary dries out first. If the boundary dries out first, then it stiffens. And then the interior starts to dry. And if the interior tries to dry, then the area decreases. The area decreases, the perimeter remains constant. Again, immediately you will get a patch of a surface which has negative Gauss curvature. So it's the same idea, except over there it's driven by drying. Over here it's essentially driven by growth. And these images essentially show you exactly what's happening. So this is the growth front, and the leaves are such that over here in the wild type, the growth is slightly less than in the middle, so it forms something which looks like a bowl. On the other hand, when the boundary grows much more, when the perimeter grows much more, then the leaf starts to ripple. And it's associated with the differential cell proliferation. Can we go beyond these beautiful genetic studies? And the answer is yes. We can do so by basically doing surgery on leaves. Nobody complains. Okay? So this is the lily from here, from Southeast Asia. And the lily leaf has a typical negatively curved shape. And if you take a razor blade and you slice the leaf into strips, the first thing you notice is that the midrib straightens out. And not only that, all the slices which are curved in their three-dimensional embedding, also straighten out. So what is this telling you? So it's telling you that in the leaf, the edges are also growing compared to the center, and they're frustrated. And that's why they start to go out of the plane. As soon as you relieve the frustration by slicing it, every strip essentially becomes straight, and then the whole thing comes back to the plane. So what have we understood? We've understood that the morphology is a consequence of the differential strain associated with the edge growing more than the center and elastic energy minimization. Because if it was not minimizing the elastic energy, when I cut it, it will not relax. But the fact is that the whole thing relaxed. So it's just an elastic energy minimization problem, but in a geometric setting. And we can test that. And we can test that by taking a piece of foam. This is a physical model. And taking your nail and moving your nails over the edge of the foam, stretching it out. And when you stretch it out, in fact, a little bit, it forms a saddle. You stretch it out a lot, it forms a ripple. And those of you who've gone to a store to buy ribbons to wrap gifts, you've seen exactly the same thing, except that over there, one surface is forced to grow compared to the other. And how do you do this? You take that ribbon and you move it over a sharp edge like that of a pair of scissors. And so that essentially stretches one side and the whole thing rolls up. So it's the same idea. And biology invented, discovered, evolved this a few 100 million years ago and used it repeatedly. Okay? Okay, so if I want to do the mathematics of this, it's very hairy. I will simply show you the equations. We've published a little bit on this. I invite you to take a look. I'll just give you an idea of the equations. So they are a pair of coupled nonlinear elliptic equations. And the physical meaning and the mathematical meaning is actually very easy conceptually. The first equation is essentially a version of compatibility, the Gauss-Codazzi relations in differential geometry, which basically say that if I have closed curves, then I must make sure that after deformation, those curves remain closed. So it's a compatibility relation, which essentially tells you something, it tells you a relationship between the Gauss curvature and the Laplacian of the area, or related to the stress in the sheet. That's my first equation, a compatibility relation. And the second equation is associated with force balance in the vertical direction, and it's a correctly covariant form of Laplace's law, which says that the tension multiplied by the curvature is balanced by the pressure. So this is essentially a version of Laplace's law. Kappa m is the mean curvature, kappa g is the Gauss curvature, sigma is the in-plane stress tensor, or if you like, in linear elasticity, is just proportional to the metric tensor, and lambda g and omega g are two scalars that you construct from the two tensors associated with growth. And there's a single parameter in this problem, and that single parameter is the ratio of the thickness compared to the other dimension, and it's a small parameter. So what does this basically tell you? All of shape of these two-dimensional objects is a consequence of the frustration induced by embedding a non-Euclidean metric in 3 dimensions. So I start with an object which I can't perfectly embed in 2 dimensions, and now I allow it to essentially relax. How do I allow it to relax? Based on its elastic properties, and its relaxation, depending on how stiff or how soft it is, gives rise to all the shapes of 2-dimensional objects that you see. Crumpled pieces of paper, the results of almost all my calculations, wrinkled skin, and everything in between. So again, mathematically, you essentially transition from oscillatory instabilities to singular objects by basically transitioning as a function of epsilon from one to the other. And all the shapes that you see in thin sheets can, in some sense or the other, be thought of as a consequence of this. And again, I want to give you a sense of the equations rather than get into much detail. And the way I'm going to give you a sense of them is by essentially giving you a numerical solution, in which it's very easy to motivate. We can do analysis, and we have done that, but I won't tell you about that right now. And so the numerical analysis is associated with discretizing the leaf using a set of triangles and having 2 kinds of energies. Energies associated with stretching the edges of the triangles, and energies associated with asking how strong or how weak it is to deform neighboring triangles relative to each other. So they're perfectly parallel. All the normals are perfectly parallel. The sheet is flat. As soon as it starts to become non-parallel, it costs you energy. And what drives the non-flatness? For example, if the edge grows relative to the center, then these triangles are stretched. If these triangles are stretched out compared to the center, then the sheet wants to ripple along the edge, and as a consequence, that ripple will propagate into the interior. And you can solve this problem by writing down a discrete energy and doing gradient descent numerically, or you can do some analysis on the PDEs that I showed you. I'll simply tell you that we've looked at both, and I'll just tell you the results associated, or show you results associated with numerical minimization. Okay, and again, we can essentially construct a phase diagram. We can construct a phase diagram associated with now what are the shapes of two-dimensional leaves associated with differential growth. So the horizontal axis is how much the edge grows compared to the center. And I'm thinking about very long leaves. And the vertical axis is the ratio of the only two length scales for a long leaf, which is the width divided by the thickness. Okay? So if I have very little growth, the edge is not growing very much, and if the width is very, very narrow, if the leaf is narrow, it's flat. So in this region, the leaf remains flat. As I increase the growth along the edge, I transition from a flat sheet to a saddle. And you can immediately see how a saddle allows you to accommodate growth, because the rim of the saddle is larger than the interior. So a saddle would be like this, and the interior would be curved. So the center line is going to have a shorter length than the rim, which is exactly what you need if the edge is growing. And if you grow even more, then the saddle transitions to a rippled state. This straight line, these two straight lines, are the consequence of simple analytic prediction. And that analytic prediction actually just follows from dimensional analysis. There has a slope of 1/2, and that's because there is a symmetry associated with a leaf like this, with a saddle which points with this concave surface up. And if I flip it around, it's exactly the same thing. And so you get a relationship just from that argument, which has a slope 1/2. Okay. So if you can understand the morphospace of leaves, you can ask, can you understand flowers? And I'm going to use, to motivate this, a wonderful book, in fact, which, again, if you've not seen, I encourage you to look at, by the German poet, writer, and natural philosopher Goethe, called Metamorphosen, in which he essentially was the first person, I believe, to try and scientifically start making sense of the transition, potentially, between leaves and petals. And he said petals are essentially modified leaves. Of course, they have a very different function, because they're used to attract pollinators, for example, and so on and so forth. But from our perspective, our mathematical perspective, here's the question. So I told you about the lily leaf. I'm sure you've all had lily flowers in your home, and the lily flower starts out as being convex, and over a period of a day or a day and a half, it opens. So it's a convex bud, and it turns out it opens. So how can it open? How does it open? So again, from a mathematical point of view, I'm taking a surface which has positive Gauss curvature, so it's close to being ellipsoidal, and then I reverse the curvature. When it opens, it's got negative Gauss curvature. So there are two ways to do this. At least two ways. One is you grow the inner surface of the petal compared to the outer. Okay? So if you grow the inner surface has more area, and so it's going to open out. And that's the solution that everybody had thought. But nobody bothered to do the experiment. And the experiment is an easy one to do. Okay? You take a petal, and you mark it with a pen along the boundary and the interior, and then on the outside, and then you see what happens. And in fact, I've already sensitized you that there is one way, which is to grow the inside compared to the other, but there is another way, which is to grow the boundary. Okay? And when we did the experiment, we, in this case, is Hai Liang. First, I want to show you time lapse. Okay, so this is over a period of about 4 hours. And you see its reversed curvature. It went from convex, positive Gauss curvature, to concave, negative Gauss curvature. And look at the most beautiful thing associated with the leaf, petals, are the edges are rippled. But I've already told you how to ripple the edges. So in fact, the obvious solution to grow the inside compared to the outside is not what the petal does. What happens is that the edge grows. Okay? How do we know that? We can do experiments, very cheap experiments with a pen, a cheap camera, and you take pictures, and you do surgery, and nobody complains. And so we can measure it, and so that's what we find. And then from a mathematical point of view, it's also equally beautiful. So these are, I didn't get into the details, but the equations are elliptic. And elliptic equations have a maximum principle. And in fact, these equations, because they are fourth order, don't quite have a maximum principle. But nevertheless, in elliptic equations, you know that I can achieve control by controlling the boundary. So biology has sorted out and figured out that, in fact, to control the shape of the object, the most natural way is to control the boundary. And from an energetic point of view, it's also very powerful, very beautiful, because by controlling the boundary, I don't pay a huge cost, because I'm only paying a cost associated with line length rather than area. Okay? So this suggests that perhaps there may be engineering applications and implications of this, which again have been around in biology, as I said, for 100 million years or more. Okay? And there's a simulation of that. Okay, so I'm going to now, I've told you a 1D problem, I've told you about a 2D problem. What about slightly even more different and perhaps even more complicated problems? Can we essentially use the same idea? And I'm going to now tell you, well, I've sensitized you now very strongly to how differential growth can lead to shape. I want to tell you a third example, which is the example of how your gut formed in your mother's womb. Okay? Of course, we don't do any of these experiments with humans. These experiments are done with chicks in a very close collaborator's lab, Cliff Tabin, in our medical school. And so I want to orient you. So this is the chick embryo, 3 days after fertilization of the egg. The chick hatches at 21 days. And so in the next 12 days, from day 3 to day 15, this is the gut. This is what eventually will become the spinal cord. That's the forebrain, which will form, and part of it will essentially become the eyes. So the gut, so I orient myself, so I'm looking like this, the head, the gut over here, and the gut is initially in a mid-sagittal plane, and it's symmetric. Okay? So it's a simple tube, which essentially connects two holes, and that simple tube is in the plane, and then at some point, about day 4, day 3, day 4, it starts to bend out of the plane. So it essentially loses left-right symmetry. And in fact, there's a beautiful question. Just like in physics, we've thought forever about symmetry breaking. Also in biology, the same kinds of questions appear. Okay? So it breaks symmetry in the mid-sagittal plane, and it tilts to one side. So you can see it's just beginning to do that. Then it forms a part of the pitch of one helix. It now creates something more complicated, and over a period of about a week or slightly longer, you form these looped structures. And this looped structure is not perfectly, but reasonably reproducible. So these are two chick embryos which show what happens. Okay? And that's a blow-up of the same thing. So in about 12 days, the gut grows from being 7 millimeters long to more than 20 times its length. But the body doesn't grow that much. So the body grows much, much less. Okay? And so this is an interesting solution to a packing problem. So the gut is long, but it has to be accommodated in a small volume. Your gut is about 8 and 9 meters long, and hopefully it remains tightly packed in the body cavity. Okay? Some of us lose the battle earlier, and some of us lose the battle later, but eventually everybody loses the battle. Okay? So how is this packing problem solved? Well, here's the solution. One solution is, it's just the body cavity. Just hold everything inside the body cavity, and the body cavity will pack this. Turns out that's not true. How do we know? You can cut the gut away from the body, take it out of the cavity, and the gut preserves its shape. So the packing problem is not solved from external constraints, by using external constraints. Another thought is that it could be constrained by this diaphanous membrane. So I didn't tell you, but now I will. The gut is not free. The gut is attached to the body at the two ends, but it's also attached to the body everywhere along it, and it's attached to the body everywhere along it by a thin tissue called the mesentery. Okay? And you can barely see the mesentery over here, but I hope you can still see it. It's a very thin, almost single-layer thick tissue. So that tissue turns out to be important. And how is it important? Well, so the experiment you can do is you can ask, if I take the gut out and I separate the gut from the mesentery, something beautiful happens. So this is the gut plus the mesentery removed from the body cavity. You cut away the membrane, the tissue which connects the gut to the body. The membrane shrinks and the gut straightens out. This is just a leaf, but in disguise. Okay? Because now think of that gut as the boundary of the leaf, and the boundary of the leaf or the gut is growing, but the interior, the bulk is not growing. So if I have a sheet and the boundary is growing and the interior is not growing, then the interior is going to be stretched, the boundary is going to be compressed, and the whole thing is going to coil up. Okay? So it's the same idea again and again and again used by biology through evolution. To see that that is indeed the case, we can do a physical experiment, just like I showed you the physical experiments previously. If you cut this one artery over here, then this coiled loop flattens out, and you see it forms this particular set of omegas. And you can, in fact, recreate this in a physical experiment with a few pennies. You take a rubber sheet, you stretch it, you take a rubber tube, you stitch it to the rubber sheet, and you let go. So the rubber sheet, which has been stretched, now wants to contract, but it's not allowed to contract near the boundary because the tube is attached to it, and so the tube starts to buckle, and the tube essentially buckles into this form. Except that there is a topological constraint. And the topological constraint is because the tube is attached to the body everywhere, it can't form a helical pattern. Okay? Its linking number, okay, so a very beautiful theorem, has its antecedents in Gauss's work, but was really formalized only in the 1950s by mathematicians Calagareanu and White, say that the linking number, the number of times that if I draw a frame and I ask how many times the frame rotated, the number of times that it rotates has to be 0 because it remains attached to the body. So every time it tried to twist in one direction, it has to untwist. Okay? And that's why this object looks locally chiral, but it's globally achiral. It looks like it's left-handed for some time, but it has to reverse and become right-handed, and then left-handed and right-handed. And you might think this is just a mathematical oddity, but in fact, there are people who have a disease associated with the problem with this called gut volvulus, and sometimes the gut can detach from the body, and when the gut detaches, then you can get an obstruction. And when that obstruction happens, a surgeon has to go in and actually operate in an emergency. And that surgeon better know topology and surgery simultaneously. So he has to basically make sure that he resects and fixes things properly. Okay, so can we now understand this? In fact, I've already told you everything now. I've told you that we can recreate it in a physical experiment. We can construct a mathematical theory analytically and predict the wavelength and the amplitude. We can do some computations. We can do experiments, and when we do experiments, we can do experiments with the chick, we can do experiments with sheets of rubber, we can do experiments on the computer, and of course on a piece of paper, and everything matches with no adjustable parameters. We measured everything, okay? So at different time points in the chick's development, 16 days, 8 days, and 12 days into development, we can measure the wavelength, lambda. This is the prediction from the scaling theory. The computations give us the prefactor. ET is the material property of the gut. I is the area moment of inertia of the gut. EM is the elasticity of the mesentery, and H is the thickness. And we can also predict the amplitude. And then you say, this is pretty good. Then you get greedy. Can you do it in one organism? Can you do it in many organisms? And so in Cliff Tabin's group, working with Cliff Tabin's group, we basically looked at not only the chick, but we looked at the quail. Quail eggs, as you know, are very small, but I'm caring about shape and size are different. Size is the 0 mode of shape, if you want. Then we looked at favorite of evolutionary biologists, finches, okay? And then finally, to challenge the genetics, I put that in quotes because this is a physics, we've reduced it to a physics problem, so there should be no influence of the genetics at this level. We looked at the mouse, and every one of them, we measure these properties, and all of them fit on the same predicted curve or straight line in the scaling plot. So that gives you some confidence that, in fact, at this appropriately coarse-grained level, things do work. So how much time do I have? About 5 minutes or so? So 5 minutes, all right. So, all right. Again, you see this idea everywhere. You just become sensitized to it and you see it everywhere. You see this in a sea anemone, in the mesentery of sea anemones. A graduate student working with me on understanding aspects of insect wings showed me recently a picture of a dragonfly wing, and the boundary is similarly shaped. I showed this to our daughter a few years ago, And then she went into a shoe store and showed me a very beautiful pattern associated with lace, where you see exactly the same thing. Okay, so you see this repeatedly all over the place as soon as you're sensitized to it. And I've told you from a physical point of view what this is about. I think it's worth pointing out there's a very beautiful analog to an old classic problem in geometry, which is associated with embedding surfaces. Goes back all the way to Riemann, was first solved and made Nash very famous in the 1960s, and then Gromov basically lifted it and created this whole field of convex integration. And in a nutshell, the question is, if I have a manifold of m dimensions, which I'm trying to immerse in a manifold of n, how large does n have to be compared to m so that I can do it smoothly? And Nash basically said and showed that if I give up continuity of a certain amount of a certain degree, then I can do this without almost any problems at all. And so what biology is essentially showing is that it's able to do this. It gives you a constructive approach with a physical realization and an energy. Rather than thinking about it purely as a mathematical entity, it's saying that if I introduce an energetic framework, these wrinkles or folds or loops are essentially a way of regularizing the problem, which in the Nash-Gromov construction would give you an iterative infinity of solutions which essentially approach the smooth embedding problem. Okay. So what have I done so far? So I won't probably have a chance to tell you about the brain, but I want to finish by telling you a little bit about the rest of it in the context of the intestine. So I've shown both in the context of, or in three cases, the shoot, the leaf, and the gut, that given the growth patterns, we can predict morphology. But there's an equally interesting problem which is associated with the feedback from morphology back to growth. Because differential growth gives rise to forces, those forces give rise to shape, but the shape feeds back on the growth, because otherwise we wouldn't end up where we are. We just keep growing, and potentially that will have chaotic, not to say fatal consequences, okay? So two kinds of questions. One is, can you essentially think about feedback? And second, if you turn your head sideways and say, oh, I'm not interested only in the physics or the biology problem, I'm also interested in a technology problem, which is, can we design growth patterns so that we can start with a given shape and end up with any shape? How can we solve the inverse problem? It's an inverse problem now in physical geometry, if you want. And I won't have a chance to tell you about this, but I at least want to raise the question. And I'll stop with this example of understanding the feedback now from growth, from stress back to growth. So how can we control differential growth? So again, continuing work with Cliff Tabin's group, we asked whether we can essentially change the growth patterns. And in a nutshell, what their group has shown is that you can use retroviral techniques to change proliferation patterns. And the way you do this is by changing the expression of a particular diffusible signal called BMP2, bone morphogenetic protein 2, and its antagonist NOGEN. Okay, so this is the wild type gut. The only thing that you need to pay attention to are these red dots, which tell you how often the cell is dividing. So more red is more division. So this is the gut. So gut is the tube. This is the mesentery, the membrane which attaches to the body. The body is not shown over here, so there's the body. And this is the wild type morphology. Okay, lot of division in the gut because the gut is growing compared to the mesentery. So if I have growth, then it's stretching, it's put into compression as a consequence of being attached to the mesentery. You change the expression levels of this protein, which changes the proliferation, and you compare these two figures, and what do you see? The number of red dots over here is reduced, so proliferation is reduced over here, but you see that it's almost gone from the mesentery. Over here, it was there in the mesentery, it's almost completely gone. Okay? So what does that mean? That means the differential growth has gone up. Okay? The differential growth, because the mesentery is not growing at all, while the gut is still growing a fair bit. If the differential growth has gone up, that means the stress on the gut is more. If the stress on the gut is more, that means the gut should coil up even more. And that's what you see. Same scale, you see the gut is now much more tightly coiled. It's also more disordered, but the reason it's more disordered is because it's very hard to essentially do this uniformly. What if you now use an antagonist to BMP, NOGEN? Again, I'm not going to get into the biochemical details, but if I have an antagonist, I expect that I will be able to increase the growth over here and in the mesentery. So if I change the differential growth pattern so they're almost the same, what do you expect? Less differential growth, less stress, larger wavelength. And that's exactly what you see. Okay? So you see now the red dots are much more uniform in the gut and the mesentery, much less differential growth. Much less differential growth causes the whole thing to stretch out completely. Okay? So here is the molecular circuit. So I've told you everything top down. Now we can start going bottom up. BMP2 expression changes the differential growth by increasing the differential strain, and therefore the loop size decreases. That's that part. Noggin inhibits BMP2, decreases differential growth, and the loop size increases. And you can make sense of this mathematically by essentially measuring the properties. And when you measure the properties, you essentially find that you can essentially predict things. So what is this telling us? So it is telling us now, not only can I essentially use physics and a little bit of mathematics to essentially predict the pattern, but it also tells you biologically there are knobs that you can tune, and these knobs allow you to move through pattern space. And so the grail over here, we aren't there yet. The grail would be, can we start addressing this question from an evolutionary point of view? Because we do know that gut shape and gut length change when you go from one organism to another. Okay? Carnivores have short guts, herbivores have long guts. In fact, in herbivores or in omnivores, you can change the gut length by changing the diet. In some rodents, you change the amount of changed diet, you change from a carnivorous to a herbivorous diet, and the gut will increase. You change it back to a carnivorous diet, and the gut will decrease. We aren't there yet, but these are clues which say perhaps by tuning very simple things like cell proliferation rates, we can change morphology. Okay, so I think I'm out of time, so I won't tell you about what happens inside the gut because it's exactly the same story again. I will instead stop by also not telling you about our ideas on how we've been thinking about the inverse problem. I'll only tell you about the forward and summarize by saying what I've shown you is a very, very simple idea that differential growth allows you to essentially understand morphogenesis of one-dimensional objects. The work was done primarily with Raghu Chellakot, who is now a professor in IIT Bombay, physics. The work on the leaf was started with and continues with Hai Liang from USTC. He was a postdoc with me. He's now back there. He's a professor of mechanics. And the gut work has been a long, ongoing collaboration with Cliff Tabin's group. In our group, started by Thierry Savin, who did all the biophysical experiments and started the computations, and they were picked up and continued by Thomas Tallinn. Thank you very much.
Yeah, it's a very nice talk. So I want to start with a question about the setup of your framework seems to me is based on the force balance and all this
and mass balance.
Equilibrium
Yes.
process.
Yes.
But for the growth of the cells and differentiation of the cells, that's really driven by this myosin, right? So we consume ATP energies.
Yes.
How this non-equilibrium energy pump into your system, build into your model?
Yeah. Okay. So that's a great question. So where do non-equilibrium processes come in? All right. So I deliberately chose to tell you about problems where shape is not associated with differential flow, but with differential growth. And so if I'm thinking about differential growth, then what matters is that I've added or subtracted material differently at different locations. Because I'm thinking about differential growth, then at short times, what you're saying is true. I will have these expression levels and or contractility and or even flow, which is going to be important. But at long times, that's not important at all. And so if I ask then how we have been thinking about these problems, at every instant of time, I'm in elastic equilibrium. And so the elastic equilibrium is quasi-statically changing over long periods of time. And so in this framework, I don't have real dynamics ever coming in, because at every instant of time, I'm at equilibrium. There are other problems that we've looked at, which I didn't tell you about today, for example, associated with how the body elongates, where I have to worry at all times about the flow. And in those situations, I have to write down dynamical laws rather than equilibrium laws. And then you can ask, how do you know? And right now, it's an episode by episode. It's a case by case. Sometimes the system behaves more like an elastic case. I chose to show you examples where that's true. But other times, it seems to behave more like a fluid. And sometimes it essentially switches from one to the other. Is there a general framework? I wish I could say yes. Unfortunately, I don't know of a general framework, except basically you have to look at every system individually.
So in your the first part of your model, you have this diffusive length for the growth tip.
It's a growth zone, yes?
Growth zone.
Yes.
Is there any physical meaning for that zone?
Yes, that's
Why
the zone.
like that?
So that's the zone, continuing along the thought that you had, that's the zone over which the cells proliferate. In the rest of the tissue, the cells are not dividing anymore, they are lignifying. So the cellulose is basically gradually being converted to lignin, which is very, very heavily polymerized polysaccharides. So in the part which is green, so if you go outside and you look at growing shoots or growing leaves, the part which is green, the cells are still proliferative, very much more flexible than in the other parts. And as they divide, and they divide only in that zone, and if they keep their shape the same, then the length will change. If they change their shape, then their aspect ratio of the tissue itself will change. So does that help?
I do have a comment. You mentioned about the herbivores typically have longer guts than, I think I read somewhere saying Asians have longer guts than Caucasians.
I didn't know that.
Yeah,
Okay.
there was, because we eat more vegetables.
Which way is it? You said because we eat more vegetables, are you sure that it's causal?
I don't know. Yeah. The other thing you mentioned is, a surgeon better know some topology and mathematics. I remember Roger, when he was here, Roger Brockett was our mutual friend. He was a professor at Harvard. He said he had a student, actually, who's currently a professor in applied math somewhere in California, but 2 days a week, he's also a surgeon. Do you remember that?
No, I didn't know that.
Okay.
So very interesting. I mean, the curvature stuff. Is that also true for hair?
Yes.
So, but hair does not have an attached sheet. So is it, I mean, how does it form the, like, curly hair? Yeah.
Because hair is made of keratin, and the keratin is essentially extruded out. So you have one cone sitting inside another. But the cross-section is different for different kinds of hair. So for example, a typical Asian hair has a larger cross-section, but it's approximately circular. Hair, at least that I am familiar with from people from the middle part of Africa, the cross-section is not circular, it's more elliptical, and it's slightly smaller. Caucasian hair is circular and still smaller, which then explains why Asian hair, when it's very small, is very straight, because gravity is not essentially playing a role since it's too small, so the gravity elasticity competition is not kicked in. But when it's long, it falls very beautifully because it's now very heavy. And with Caucasian hair, you don't see that. And if you have a cross-section which is elliptical, then it's very hard to keep it straight, and so it starts to twist up. Okay, I was once a consultant for L'Oreal. But I don't have any hair, probably that's why they felt very safe working with me. But they refused to allow us to publish anything, so I stopped.
Okay, Michael.
Can you tell us what determines the shape of the egg?
Oh yes, I can give you an attempt at that. So first of all, it's not the shell. Everybody might think it's associated with the hard shell. And here's an experiment, again, for you to try with your children or nieces or nephews or what, take an egg and put it in vinegar. I'm sure people actually consume eggs in vinegar. If you put it in vinegar, the vinegar will dissolve the shell, but the shape won't change. It will swell a little bit, but the shape won't change. So it's got nothing to do with the shell. What we know is that the egg forms in the oviduct, and as it moves through, it starts to grow. So it's under pressure. The membrane, which is just under the egg shell, actually is not got uniform thickness. So in the zone which is pointed, the membrane is thinner, and in the zone which is flat, the membrane is thicker. So you break symmetry by essentially having variations in the membrane thickness. So now you can think of the egg as a pressurized balloon, if you will, except that the balloon is got a thickness which varies. If the thickness was uniform, you get a sphere. If the thickness varies, you essentially get the shape that we have. We measured, or rather somebody else had measured, and we took the measurements of the membrane thickness, and we constructed a theory for it. What we haven't done, as far as I know, it's surprising. I think I once estimated that the number of eggs consumed by humanity in a year is of the order of a few 100 billion. Okay? And nobody has observed how the egg comes through the oviduct. Does it come out pointed side, or whether it comes out blunt side? Okay? Why might this be of interest? It might be of interest because there is another possibility. So, as I said, you break symmetry. So when you break symmetry, do you break that symmetry early on, and then you just continue? Does it move out with the pointed side out, or does it move the blunted side out? I don't know. So we tried to get permission to essentially do small animal X-ray studies. We haven't gotten around to it yet. Maybe we will at some time. But in terms of the membrane thickness variations and the total volume, we can predict that phase space that I showed you. And the reason that one corner is not occupied is because that one corner will require membrane variations from the one pole to the other to be extreme. And we see no reason. We have never seen eggs which have that. So perhaps that's why you don't see anything in that corner. Sorry, long answer to a simple question. Even more beautiful question is, who came first, the chick or the egg? And there is an answer. There is an answer, which is perfectly sensible.
What is the answer?
Well, what do you think? Did the chicken come first or the egg come first?
I will say chicken.
No, the egg.
Oh, okay.
And here's why. Because the egg predates birds. Because the egg was a spectacular. Yeah, exactly. You can go back to dinosaurs.
Yeah.
So the egg did come first.
Oh, yes. Okay.
I mean, it's not a trick question.
Right.
I mean, it's not a trick answer. It's
That's very good. Yeah. Any other interesting questions? We touched on guts, hair, plants, and what else can you think of? Any other questions?
Thank you very much. So you mainly talk about this process from, you could say, growth to form.
Yeah.
And I guess an example of inverse process could be prion protein.
Yeah.
So I was wondering if you have any insights or comments on that problem.
We've just started asking questions about what misfolding might do. And how misfolding gives rise to complex shapes. We haven't thought about individual proteins, but we've asked how the aggregates form complex shapes. And in a nutshell, if I have fibrils of individual beta sheets, which are essentially starting to bind together, you have two kinds of interactions. You have interactions along and across. And if these interactions along the fibril and across the fibril are different, then it turns out you can get complex shapes. But I'm happy to tell that to you offline afterwards, because it's a little bit complicated.
Okay, if not, let's thank the speaker again.